Dr. Walter Koroshetz, Director of the National Institute of Neurological Disease and Stroke at NIH. Dr. Koroshetz trained initially at Georgetown and took his medical degree at the University of Chicago, did neurology residency at Mass General, postdoc at Harvard Neurobiology, worked with David Corey and Ed Freshpan, worked on excitotoxicity, and then went on to make a number of advances, including MR imaging and acute stroke care, chaired neurology in the stroke section at Mass General, where he was professor of neurology at Harvard, and then spent about 8 years as the deputy director of NINDS until last year being appointed as director of NINDS. He's been responsible for a number of major initiatives, including co-leading the NIH Brain Initiative. I'm glad to say he's also a friend and a good visitor, and we've had the opportunity to invite him to a variety of places that he's willingly shown up, so it's a pleasure to have Dr. Koroshetz back. Please join me in welcoming Dr. Walter Koroshetz.
Well, Mike, it's a great pleasure to be here. It's always good to get out of the Beltway and come down to some pretty part of the country and to see some old friends and meet some new friends. So I'm probably should have came before Carl because he basically kind of hit all the high points that I'm going to be talking about. But just let me put it in context a little bit and maybe build on what he talked to you about. But I think the challenges for the future were in his talk. Okay, so to start out with, I just want to make a couple of points about neuroscience. And the first one is that there is tremendous interest not only in the scientific world but also in the public view about how our brains work. And I think at some point somebody's going to find a circuit which is in all our brains, which is put together because it's there to force us to try and understand how our brains work. What this has done is, on the science world, it has brought many people into neuroscience. And so if one looks at the number of PhD students and going into different areas of science, they're all flat except for neuroscience. Neuroscience is going up exponentially. It's also the number of PhDs going to neuroscience is about double of what's in any other field. And from the medical point of view, we really like this because the problems that we're dealing with are the neuropsychiatric disorders, which are the leading cause of disability in the United States and around the world. As the population ages, even in the underdeveloped countries, it's becoming greater and greater as well. So we have big problems on the medical side. We have a workforce that's coming into the field, and then we have these new technologies that Carol talked to you about. So we're in a really sweet time in terms of trying to make progress because of these three things. Now at NIH, the budgets are not determined by a top-down decision. The budgets at NIH, for people who submit grants know, is really related to the fact whose grants get funded. So the more people you have in neuroscience, the smarter people you have in neuroscience, the more grants you're going to get funded in neuroscience, and that drives the neuroscience budget up. And in fact, what people probably don't know is that neuroscience is the largest bucket of money at NIH compared to the other areas. The reason why most people are not aware of this is because in cancer, all the money is in the National Cancer Institute, and it has this tremendous profile going out to the country. In neuroscience, the budget's split up among multiple different institutes. So the tough news is for us to actually make a strategic move in neuroscience, we really have to play a consensus game and move the ball forward. The good news is if you're an investigator, you have lots of different institutes that you can apply to for money depending upon your fit of your science for that institute's mission. So lots of institutes. So the Eye Institute, for instance, does tremendous funding of visual cortex work. Child health and development of the brain. National Institute of Mental Health, a lot of neurodevelopment, a lot of circuit studies, and Aging Institute. The Aging Institute at NIH budget has almost doubled in the last 3 years because Congress has been extremely nervous about the growing public health problem of Alzheimer's disease, And so the Alzheimer budget at NIA has gone from about $440 million to over $1 billion in the last 3 years. Again, almost all of that is neuroscience related research. So neuroscience is booming. Now, the bad news is that the NIH budget as a whole overall has been flat. So that the worrisome thing is we have this workforce coming in. We have a pretty much flat budget with the exception of the new money that's coming into Alzheimer's research. The hope is that that will somehow break because I have been, and others at NIH talk to people in Congress, and in general, the message is actually very well received that these 12 years of flat budget at NIH have really damaged our preeminence in the field of biomedical science, and that really can't continue. We did get an increase in 2016, but that as a one-year increase is not really going to do it. And I think that the Congress has bought into that. The issue with the Congress is if they actually do a budget, and that's usually where the problem lies in trying to actually get them to move on a budget. But there is hope. Now, from our institute, I just wanted to throw this up to just give a sense of where we invest our money. What we try to do is actually not leave any area untouched. So if someone has a good idea in almost any area in the Neurological Institute, we'd like to have some funding mechanism for it. Now, that being said, it's not all equal across the field. So about 25% of our funding goes to neuroscience research that's disease agnostic. How does the brain work? How do brain circuits work? About 40 to 50% of our research budget goes to basic mechanisms of disease. So protein aggregation in Parkinson's disease, recovery mechanisms in the brain after stroke. The rest of it is divided about half and half between actual clinical trials where we're testing things in patients and translational research. At NINDS, we have actually a translational research program which can be described in a simplistic manner as a virtual pharmaceutical company within the government. It's virtual because we have maybe about 10 staff people, but what we do have is we have contracts with industry folks who are the same organizations that pharmaceutical companies contract with to bring along a molecule to a point of FDA, IND, and then into patients. So we are able to hook up investigators at universities with these contract companies if they have a good idea to bring a molecule along or a device along to get into people. So that's our translational research program. We spend a lot of time in training because we're very well aware of the fact that the future is not that far away. Time passes quickly, and it's really the young people who are coming into neuroscience now that are going to make the advances that we're going to be talking about. I'm going to spend most of my time on the Brain Initiative, and the Brain Initiative was announced by the President in April of 2013. And actually, we were quite surprised by this. We didn't know this was coming. There had been a lot of talk before the announcement. The Kavli Foundation, for instance, was very instrumental at kind of pushing the idea that it could be a grand challenge in neuroscience. There was action at the White House Office of Science and Technology and Policy, and people would come over. The congressman came to NIH to visit, and I took them on a tour. And he was very interested in neuroscience, and he said to me, well, what would it take for us to know how the brain works? And I said, well, we need all these new technologies where we could look at the animal level and connect it to behavior, to the human level and connect behaviors to neurocircuit activity. But I said, there are things that would be very, we could do. And he said, oh, good, well, I think we should do that. And I said, well, that'd be great. And I said, well, how do we convince your fellow congressman to buy into this? And then I had the most shock I've ever had. He just said to me, oh, don't worry about that. We'll take care of that. I said, whoa. But then, it happened. So he obviously had the ear of people in the White House. So remember that so that's how politics works in Washington. You want to know. But the point to make is that it was really quite interesting that the decision at the White House level was to invest in technology development, something that NIH never really did very well. We kind of putted around the edges, but never really made a big effort in neuroscience at technology development, which I think was a huge mistake, because once we did this, we have seen amazing people come through the woodwork to try to help neuroscientists understand how the brain works. Now, we're not doing this alone. The Brain Initiative is multiple federal partners. The National Science Foundation particularly works very closely with us, but also we have people from the FDA, because as we'll be talking, the Brain Initiative has technologies for experimental work, but it also has technologies that are going to be used in humans. And then IARPA and DIARPA are also investing money in trying to understand brain circuits and so and connect us with us. And then the private organizations, Genelia, for instance, as Carol mentioned, tremendous contributions. The Kavli Foundation I mentioned before has been, a real force in trying to move this forward. The Simons Foundation, Jim Simons, incredibly interested in the computational neuroscience, and as Carol mentioned, the challenge going forward is really going to be, what do you do with all this data? How do you make sense of all this data? And how do you don't go down the wrong track? And so the ideas, we need ideas on how the brain is actually, behaving, giving all the data we see. And we need to know what the rules are. Before we could see all the neurons firing, we had no chance of understanding the rules. But now we have all the neurons firing. I think what we know is, whatever we thought before is completely wrong. We have no idea what this network activity, how it's generated, how it's associated with behavior, how it changes over time. And then the Allen Institute for Brain Science has also been at the forefront in many of these areas, particularly in the classification of different cell types and how they're connected and how different cells have different properties, kind of the parts list of the circuits in the brain. So all these groups, we're actually trying to work all together. We have more meetings than you can imagine, but I think it's working pretty well so far. Now, Carol talked a little bit about the physiology issues, and I think what we have to, they look complex enough, but remember that the spatial problems, the scales that we're dealing with are also quite tremendous. So this new technique, Clarity, which allows you to take the lipid out of the brain, make the brain transparent, you can then see with fluorescent markers that you've accessed to the animal before death, and before doing this, you can see now in 3D all the kind of architecture that is making up these circuits. You can do experiments where early gene expression is expressed, and then there's a fluorescent marker associated with the cells that lit up with the early gene expression. And then you can do this and see in a 3D animal's brain where all the areas that lit up in response to a certain stimulus. Say, an experiment with Haldol, for instance, where Haldol was given to the animals, early gene expression associated with a fluorescent marker. You do this, you can see in the brain where Haldol activated in that kind of a time frame. But the focus of the Brain Initiative is really on understanding the circuits, so it's a structural and a functional side to it. What we'd really like to get at is the fluctuating electrical and chemical patterns that are occurring within circuits, and to understand how these patterns of activity generate our thoughts and actions. So that's kind of the ultimate goal. From the medical standpoint, the way I think about it is that way we have been operating, at least in neurology, is that we have been very much focused on the phenotype of the individual. The tendon reflexes are up, they're down, the toe goes up, the toe goes down. The patient has a cognitive ability, aphasia, apraxia. And then we look in the brain to see what the molecular structural pathology is, and we tie those things together. But what we've known all the time, but haven't been able to really measure it, is that the patient's problems are evidence of their circuit dysfunction, and the patient, and that's the variability that you see, which not many people talk about, is because there are molecular structural pathologies that look equal in the same brain, but they have different effects on patients because they're affecting the circuits differently. We've never been able to see that circuit dysfunction. We've been making this jump from the pathology to the patient without being able to measure the circuit dysfunction. For the psychiatric disorders, where they haven't been as lucky to have molecular structural pathology, they have the phenotype. They know that there's a circuit function problem, but you can't really get at it. And I think that again points to the importance of trying to understand circuit function, being able to measure it in patients. If we can do that, that changes how we approach our disorders, and it really makes the approach to disorders much more precise. So the patient doesn't care how much amyloid they have. What the patient cares about is how it's affected their memory circuits. We haven't been able to measure that very well. So I think it could have a really profound impact. Now, I think that also that this statement by Friedman Dyson kind of captures the way we think about the Brain Initiative going forward, and that is that new directions in science are launched by new tools much more often than by new concepts. The effect of a concept-driven revolution is to explain old things in new ways. The effect of a tool-driven revolution is to discover new things that have yet to be explained. So Carol's picture of all the activity in hundreds and hundreds of neurons is a great example. That's a tool, gives us data, and now how do we explain that? And just one example of what's coming forward. Now, for us, the old people in the audience who are in medicine, when I came out of medical school, this was the first image of brain and living people, and we were so excited to see this fantastic image of ventricles and mush around the ventricles. But look what happened with technology over the ensuing years. This is the kind of MRI resolution that you can get. Another example of the fact that what we're doing now is not that different from what's happened before in other fields, and just the impact that technology has on the practice of medicine or the practice of science. There are lots of examples going backwards. Now, to talk about the Brain Initiative and be more specific, there are basically 7 high priority areas that were put forth by a group who wrote a report to Francis Collins, the director of NIH, about what the Brain Initiative should be looking at. And that group wrote the report. It's called Brain 2025 Report. I've read a lot of government reports. I wouldn't advise reading any of them except this one. This was really a good government report. People took off their hats. They, I think, took a very scholarly view and put out a roadmap for what the Brain Initiative would be. The first step was to try and called discovering diversity, to identify and provide experimental access to the different brain cell types to determine their roles in health and disease. So Carol talked to you a little bit about some of the technologies that can access different cell types. One thing we're trying to do is to actually get a census of all the cells in the brain. What are all the cell types in the brain? How are they different? And the idea of having a computer and trying to understand the computer, you really need to know what the different parts are that put the computer together. So this is kind of like the parts list. The second one is to develop tools that can map at multiple scales the circuit diagram that vary in resolution from synapse to the whole brain. So this is a challenge. So in simple organisms, Drosophila, you can potentially do serial EM through the brain and develop over time an intricate, detailed, extremely deep map of all the connections. That's something that you really can't do in the human brain, but then there are more mesoscale ways in which you can approach that problem. You saw, for instance, the clarity technique, which shows the connections between cells. We have the connectome in MRI, which is on a macro scale, showing the connections between different brain areas. But this is our challenge, is the multiple scale problem, not only spatially from the dendrite to the interactions between close neurons, but also the nuclei interactions across the brain, the network interactions across the brain. And then the temporal scale is incredibly important. So at what level are you looking in terms of temporal scale from milliseconds up to multiple multiple time frames. And then the issue of the brain is changing all the time. So it's not just time. The brain may not care that much about time itself, the ticking of the clock, but the brain cares a lot about the activity inputs that are coming in temporally that then change how the circuits function over time. So recording from one neuron, it does one thing at one time. 2 days later, same activity, that neuron may have nothing to do with that activity anymore. It's changed. The network is still able to generate that function. Now, to do this, you certainly need to develop tools that will see circuit activity as it's going, and Carl talked to you about a lot of those. The next level is trying to get beyond purely association. So in much of neuroscience, we've had a behavior, we have recordings from the animal, we see the activity, we see the behavior, we think they're associated. And because it's occurring closely in time and space. But to get a causality, the next step is to actually develop a model based on what you're seeing, and then to test the model by intervening. So being able to intervene is also critically important to getting at causality. Now, there are technologies, Carl talked about a lot of technologies for recording. There are also similar technologies now for actually stimulating, turning on or turning off cell types. So now we have that ability to actually get a causality by turning on or turning off neurons and seeing if the model then predicts what you think it would in that instance. The fundamental principles idea, again, Carol mentioned, we're going to be collecting these tremendous amounts of data. What are the rules that govern that neural activity and how it generates behavior? How sensation generates those patterns in neuroactivity? We believe that that we don't understand how that happens, and the rules really require the type of people mentioned, the mathematicians, the computational scientists, and so a huge influx of those groups will be really necessary. And finally, the Brain Initiative is trying to look at the problem of, as the president mentioned, trying to understand brain at the level of our thoughts and actions. So some things you can only do in, zebrafish. Some things you can do in mice. There are things you can do in people. And so the Brain Initiative is approaching the problem at these multiple different experimental areas to get at really what the brain in the human condition is doing. So Carl talked to you about a number of the different techniques, particularly the calcium imaging seen here. Big problem that we have these great tools to get calcium imaging. The problem is that the optics are limiting our depth problem. Carl talked about some of the ways in which you can get better lateral exposure, but getting deep signals out is a real problem. So Chris Yu was a engineer, scientist at Cornell, went off to work to put in fiber optic cables into the whole country with industry, came back to try and work on fantastic microscopes to go deeper into the brain. So this is an example of some interesting optical work, and here's work from Hillman at Columbia, again looking at a new technology to look at a large lateral space of cortex. So you can see neurons firing over large lateral distances, particularly on a nice flat brain. There's also new sensors. So Mark Schnitzer is working on voltage-sensitive dyes, which have had problems in terms of their signal-to-noise in the past, bleaching, but again, using a lot of the techniques that Carol mentioned, has made progress, and some of these we think will come online fairly shortly. And again, so this gives you actual voltage sensing as opposed to the downstream calcium effects, which the GCamp's been relying on. And then you can do some really clever things, and you can develop fluorescent sensors for chemicals. So here's a GFP-linked bacterial protein mutated to bind serotonin. So when serotonin is released, the cells that the serotonin hits will light up. And so you can actually sense chemicals as well as electrical activity in the brain. Now, what do you do with this? Well, we have programs in the Brain Initiative called Integrated Neuroscience, where we're asking people to use these advanced techniques to try and understand circuit activity at a level never done before. Some of these things can be done in simple animals like zebrafish, where you can actually see all the neurons simultaneously with the behavior. So you can potentially, in different behaviors, identify the firing pattern, try and do the associations, try and do the interventions, develop the model, potentially have a model of the zebrafish nervous system. So starting with a primitive animal, but that's frequently how neuroscience progresses, is to work with something that you can really get your hands around the whole thing, and then try and work your way up. Some interesting technologies that are potentially coming in from outside the brain to interrogate or modulate brain activity. So this is work done at Mount Sinai, where they attach a ferritin molecule to a trip channel, and with radio frequency waves, move the ferritin, open up the channel, and then activate, or with a particular other channel technique to actually close the channel, so they can actually activate or deactivate cells who have genetically modified this trip channel. They've used this in animals in the hypothalamus to turn on and turn off glucose regulation cells and see glucose changes in the animal themselves. Some physicists have controversial issues with whether this is possible, but it looks like they've been able to do it, so we'll see, but we will see. Focused ultrasound, people may know the University of Virginia is a very focused effort on focused ultrasound. They have projects where they can put a 1 millimeter focus of ultrasound energy into the brain and create a lesion, and that has, I think, recently been approved by the FDA for treatment of thalamic tremor in people, working on it in Parkinson's disease. The interesting thing is that at levels before you cause lesions, the ultrasound has other effects. It opens up the blood-brain barrier, so you can potentially access with the blood through the bloodstream particular areas of the brain singularly, as opposed to the drug getting to everywhere in the brain. More interesting to me has been the fact that there's some evidence that at some energies, the focused ultrasound can either turn on or turn off neural signaling. So Frank Yolez at the Brigham, who is one of the main technology developers in this area, has shown that a focused ultrasound on the trochlear nerve actually can cause the eye to rotate, which is a sign of trochlear nerve firing. So wouldn't it be amazing if this technology is safe enough that you could go into people with depression or schizophrenia and search, with a hypothesis-based approach on what are the effects of turning off or turning on certain areas of the brain with focused ultrasound. Again, far-fetched a little bit, but maybe not that much. In terms of neurorehabilitation, there are projects that are looking at the plasticity of the brain and how one can intervene. So there are patients who undergo epilepsy surgery or who undergo deep brain stimulation. So they have electrodes in their brains, and they can have, those brains can be recorded from with now more and more sophistication. So hundreds of neurons at a time, perhaps. The Brain Initiative is people are developing new electrodes that can be used to get finer and finer recordings from patients who undergo these neurosurgical procedures. And they're very interesting patterns that have been looked at now, say, with just reaching tasks, and trying to understand from the firing patterns of hundreds of neurons simultaneously with a reaching task, what is the activity pattern that generates the movement. So something similar to what Carol showed, but actually in people. People are also looking at intervening. So this group at Cornell studying thalamic stimulation in people who have traumatic brain injury, who are in a minimally conscious or vegetative state, trying to move them out of that state to a more high-level conscious state, have shown some very interesting progress. And again, the Brain Initiative is funding them to kind of move this forward. And for time, am I out of time? Yeah, okay. So let me finish with this one. And this is just an example that I use to indicate that the great thing about technology is that you know it's going to do great things, but you have no idea where those great things are going to come from. So we put this together and we say that Congress is going to be able to do X, Y, and Z, but in actual fact, it might do X, Y, and Z, but it's also going to do A, B, C, and D. And so here's an example. So this was a cell census project to Arnold Kriegstein, trying to look at all the different cell types in the fetal brain. Again, that cell census project, the cell parts project. And he was able to do that. He defined a population of cells called the radioglial cells, which give rise to other cells that will form neurons in the cortex. And then what he also found, but the transcriptomic analysis that classified those cells also had a receptor for the Zika virus. So we think that this is actually how Zika is causing microcephaly, by coming in through the receptor, which is expressed in these radioglial cells, killing them off, and then you don't have the cortical cells to go and develop the cortex, so you end up with microcephaly. So again, something that you wouldn't have predicted from the beginning of the Brain Initiative, but that's what technologies really do for us. So in ending, I'd say we have the workforce. We're bringing in engineers, computational scientists, neuroscientists. The problems are great. The future, however, looks incredibly bright. The question, I think, is not whether we get to the answers, but just how long it's going to take us. So thanks very much. Pleasure to be here.